So now we have Dr. Xia Yang. She's a professor of integrative biology at UCLA, and she's joining us to discuss how she dissects complex human diseases through big data bioinformatics and systems biology. Dr. Yang is currently a tenured professor in the Department of Integrative Biology and Physiology and a faculty member of the Institute for Quantitative and Computational Biosciences at UCLA. So Dr. Yang, please take it away.
Thank you so much, Diana. I really appreciate the opportunity to share our science here. And this is really a very enjoyable event. I'm listening and seeing the very interesting questions and learning new science. It's a really awesome opportunity. Thanks again for inviting me here. So yes, I'm going to share some of our work on big data informatics and system biology, and using that to understand the complex human diseases. Okay. So I'll first go through the multidimensional complexity of human diseases, so that that build the foundation on why we even need to use this system biology approach to understand the complexity. And I'll then introduce some, briefly some work we have done in this area to learn about the complex diseases using the approaches. And lastly, I just wanted to highlight the importance of bioinformatics and big data analytics in resolving complex diseases. Okay, so first of all, let's go through some of the complex diseases we're talking about. Many of these complex diseases I'm showing here are the leading causes of death, like heart disease, cancer, stroke, Alzheimer's disease, diabetes, etc. Right? So this is the data in the US. In the world, some of these diseases are also leading causes of death. So in my lab, well, among this listed here, or we study heart disease, stroke, Alzheimer's disease, and Alzheimer's disease because they have a lot of shared etiology. Okay, so if you look at the prevalence, just take heart disease as an example. This is a US map. And you can see that it's quite, actually 1/3 of the population has heart disease. But it's more severe or more prevalent in certain places than the others. So that explain that, kind of raises the question why, and why do different people at different places might have different risks. So anybody wanted to see why that could be? Any guesses? Nobody want to guess? Oh,
socioeconomic factors.
Socioeconomic state, yeah, yes. And also environment, diet, lifestyle, right? There are many things that could be involved. So indeed, that lead to, the complexity, right? Not everybody will carry the same kind of risk. There are different types of risks are involved in the complex diseases. Like we talk about genes a lot, definitely all these complex diseases have very heavy genetic component, and also the environment, right? Depending on the diet and the environment and socioeconomic status, there are a lot of things that could contribute to these diseases. And then, so for the heart disease here, I'm showing that the heart, actually the most common type of heart disease is not about the heart muscle, it's actually about the coronary artery, which are the arteries that supply the nutrient and oxygen to the heart. And the main cause is the blockage of this artery. So where does the blockage come from? Actually come from the liver, adipose, and actually the brain can also regulate the functions of this artery. So actually it's a lot more complex than we thought. The heart disease is just the heart. It's actually involving many other different places as well. So that brings to the second complexity that, not only there are many different causes and genes and environment, but also there are many different tissues and organs that could be involved in one disease. So here I'm showing you another disease, type 2 diabetes. So for that disease, brain, liver, muscle, intestine, even the fat tissue, they can all be involved in how our body regulates glucose control. So you have to realize that we can't just study one particular, organ or tissue. Most people think, okay, it's pancreas is the problem for diabetes. That is one of the main causes, but actually there are many other things involved as well. So that's make it a little bit more complex, right? So even within each of these tissues or organs, there are many different types of molecules. So we call them like omics. So I will introduce all these different layers of omics a little later. So there are different molecules. They actually interact in networks that are within tissue or also between tissues. So different molecules interact and regulate the tissue function, and also they can crosstalk between tissues. So the key here is actually there are main key regulators that could be more important than the other ones. The idea is how we can take all the data, all the information, and find this kind of networks and key regulators. So classic approaches to address these diseases, I think you already got a flavor. We can study one gene at a time, right? So you can you can do this kind of a study by knocking out a gene or adding the gene back. So for example, if we think about type 2 diabetes, I talked about all these tissues are important. And let's see, okay, what about I believe gene A is important, let me take it out or add it back, or gene B is important. We can do this, right? We can do screens. That might be easier to do it in a smaller organism like Drosophila or worms, but actually in mice or bigger model organism, it's harder to do. It takes forever to really understand that. So actually they makes it, if we, the idea is if we understand what each gene does in each tissue, then we can piece things together. But when we piece it together, it may not be exactly what we want it to be. So the challenge that we're facing with over the past few decades is that this breakthrough discovery for so many more genes for each of the diseases, the complex diseases we talk about. If you want to take a guess, what's the range of genes that you would guess that's involved in each of these diseases? Like in tens, in few, or hundreds, or thousands? What would you say? Anybody want to say something?
Tens.
Well,
Hundreds.
actually, yeah, hundreds. Yes, at least hundreds that reach statistically significant cutoffs, right? If not thousands. As we increase the study population, we find thousands or even more. So that's what's making it challenging. Do we want to study, knock out every single gene and study every single one of them in every single individual tissue or cells? So that becomes a little bit inefficient. So so we kind of start thinking about a different way of looking at complex diseases. So this is a paper published in Cell in 2017, which bring out this concept of Omnigenic disease model, which means that most of the genes are involved in these complex diseases. Because the genes are interacting in networks, and actually the network, the roles in the networks are not all the same. Some are more important. We call them core genes or hub genes, and some are a little bit less important. They're in the peripheral, so we call them peripheral genes. Okay, so if you have to take a guess, which genes are more important, the core or the peripheral?
Oh, everyone's typing in the chat.
Yeah, everybody says core genes, great. Yes, exactly. So I think that kind of highlight our approach, right? So we don't really need to understand every single gene what it does. If we could really look at how they interact, we can prioritize certain core genes. Maybe that's more effective in looking at the disease. So okay, so I think that's my long background introduction on the multi-dimensional complexity of the human diseases, kind of pointing to some ways of we can do this kind of research. So this brings to the second bullet point here. What is system biology? How can it help us address all that multi-dimensional complexity we talked about? So this is a big data science. It's highly interdisciplinary, involving engineering, mathematical, statistical, computational principles to address biological questions. So you need to have some background in different areas to really be able to effectively do this. We focus on complex interactions, not the individual genes or component, but the complex interactions within the biological system. So we call it a holistic approach. So over the past decade or so, there has been very strong debate on the, which way to go. Do we, need to stick with the reductionist approach? Do we need to really move all the way to the holism approach? And the debate is still today. Actually, in fact, yesterday I just had a big argument with a big professor at UCSF because we're on the two different spectrums, and he thinks the approach that we're taking is making things more complicated, and that you will never solve anything, right? And my approach, my answer would be, well, we probably need to do both, right? So what I think is, if we do have a very strong belief, we wanted to find something, yes, the reductionism approach is very good. You can go really find your gold if you believe it's there. But the system biology approach is, I don't know. It's so complex. I just wanted to use the data to tell me what might be more important, right? I first look at the whole picture. I find something that's probably more important. Then I go back to the reductionism approach to really go find what I need to find. So to me, there shouldn't be debate. There should be just a harmonization of the different approaches, and we work together. One big picture, one a little bit smaller, kind of a more focused study. So put it in a bigger concept. So we will see that the system biology is just using networks of networks to delineate the complexity. So we really wanted to understand the molecular networks, all the genes, how they interact. And then there are many cell types and how the cells communicate in tissues, and also how the tissues interact to really understand the complexity. So that's my introduction for the system biology part. So next I will go through some of the studies on what we have learned about the complex diseases. So in my lab, we partition the diseases we study into two camps. One is the cardiometabolic side, so the heart disease, diabetes, obesity, etc., because they are highly connected cardiometabolic issues. And then the brain disorders from Alzheimer's disease, ALS, etc. So we also look for connections between different systems as well. So the approach we take, obviously we need computational big data integration. 70% of the lab does that. And we do have a wet lab as well, because I do wanted to integrate the reductionist approach to really, once we narrow in onto some strong candidates, we can do experiments and really test them and see if they are real, right? And then the approaches we take, obviously I have introduced earlier. So we take the different type of risk factors from big data and look for their effects on individual tissues, and go into the details on the different omics and look at how they interact and look for key regulators. All right, so here is the slide I wanted to go through these omics domains. I think for you guys, you must be very familiar with the DNA that goes to RNA, right? Goes to genes, and from genes that goes to the protein. So basically these three layers should be very clear to you. What I'm adding a little bit here is the epigenome, which is, the DNA sequence where we inherit them from our parents. But actually there could be epigenetic modifications like DNA methylation, histone modification. I'm not gonna go to too much detail, but there are different ways to modify the DNA. So you can turn different genes on and off, okay? And then the metabolome side is just small molecules like different fat molecules, amino acids, sugar molecules. There are different molecules that could fill in the molecular space. So there are intrinsic interactions and workflows across the different molecules. We can leverage that to understand the complex system. And there are also many, and I put the numbers here to just give you a hint on how big the data could be. So it's not like you can open the file with an Excel sheet, right? So it's going to be quite big data. So then from each layers of this, you can look for disease association. For example, I wanted to know on our DNA, we inherited from our parents, which pieces of the DNA is associated with the disease. So that would involve this so-called genome-wide association study, GWAS, that have become very powerful. And then if you look at the epigenome layer, you can do epigenome-wide association study called EVAS. And I'm sure you guys are all very smart. You can guess here. If I look at the transcriptome, what would that be? Need to PVAS. Yes, great. So PVAS, that's very good. And then you can go continue with that, and then protein PVAS, etc. Right? So you can really just look at what molecules are associated with the disease. Then the next task is, how do I piece all these things together to really understand the disease? Then we need to develop tools, right? So our lab, through the efforts of a few former postdocs and the PhD students, we have built this tool called mergeomics. So merge-omics, right? Try to merge the different omics domains to really look for what kind of networks are perturbed by the different type of molecular signals. How do they interact in the network? And then find the key regulators. So we have our package you can download if you do coding. You can also use our web server, so you can upload your data and do your analysis using our tool as well. And that could help. What mergeomics does is it can help us narrowing onto disease networks and the key regulators. So we can also use this kind of network to ask, can I find a drug, an existing drug, for example, FDA approved drug, to see whether that drug, any drug can perturb this disease network, right? Then we can do drug repositioning. So to do that, we developed a second bioinformatics tool called PharmOmics. So here we wanted to take all the FDA approved drugs and look for their molecular signature and in different species, in different tissues. So that way we can build a database to see, okay, a drug can affect these genes in the heart, in the human, or versus the other species. And then once we have this database, we can ask the question, if I have a disease network, so the genes highlighted in red here are the disease genes, and if I can find a drug molecule that hits the similar network, not necessarily the same set of genes, maybe that drug can be used for that disease as well, right? So that's kind of the concept we have here. Again, we have online tool that people can use. So over the years, we have been applying this kind of tools to study many different diseases. I'll just give a very quick example on fatty liver disease that we have recently did. So fatty liver disease is a very significant risk factor for heart disease, diabetes, etc. So it's basically fat built in the liver, and then it causes inflammation, causes scarring of the liver. And the mechanism is not very clear, and there is no FDA approved drug to treat this condition, okay? So we still need to better understand it. So we collected a lot of omics data from collaborators, public domains, to try to model the network. So here we modeled two different tissues. So we included both adipose and liver because we believe those are the relevant tissues for fatty liver disease. And indeed, we find many different genes in many different processes seem to be very enriched for genetic risks of the liver disease. So in adipose, you have genes in insulin signaling, for example, in immune system, metabolism, etc. And there are also processes shared between tissues. And then we take these genes, we can really look at their interaction. For example, here is the liver network we modeled based on the genes that are involved in fatty liver disease. We could predict, therefore, key regulators seem to be really important in regulating all the rest of the genes. And now let's go to do experiment, right? If I take them, lower their expression level in the liver, do they really affect the liver buildup, the fat buildup in the liver. Indeed, by lowering the expression of each of these 4 genes, we could lower the liver fat. And we also validate that when we take these genes out, indeed the neighboring genes in the network were all changing as well. So we could validate the network structure and also validate their function in fatty liver. And by the way, 2 of these are completely novel, which were later confirmed in additional human studies, okay? So that's kind of the power of this kind of approach. And another way of using the network, as I hinted, we could also look for drugs, existing drugs that target this network, right? So using the FDA approved drugs, using our PharmOmics tool, we predicted a few drugs which may really reverse the fatty liver. So we treated the animals. So here's the control mice that don't have increase in the liver fat, And then we treat, this is the disease animal that have increased fat, liver fat. We treat with one of these drugs, it actually significantly reduced the liver fat. The other one didn't work as well. It was a it had a much lower rank in our prediction. So actually the rank in our prediction also seemed to matter. So that's another way of using the network to look for kind of potential therapeutics. So that was the example for one disease. We can also study, the relationship between highly connected disease like heart disease and diabetes. We know that they segregate together, but are there shared mechanisms? Again, we can collect a lot of data from the public domain or from collaborators and do our modeling. And again, we can find sophisticated networks, but there's a structure. So there are key regulators that organizing these disease genes in different tissues. So this part of the network is mostly coming from the fat tissue. This is mostly from the immune system. This is mostly from the hypothalamus of the brain. So this again raises new hypotheses so that we can go back to the lab to test how they are relevant. So that's another example. So the last part of the example I wanted to show is we are currently moving beyond just the tissues, because each tissue, there are many different cell types as well. For example, the brain has neurons, many different types of neurons, astrocytes, oligodendrocytes. So, but when we look at the tissue, we're grinding everything together, right? So now we have the technology to look at all the genes in every single cell. So we could do thousands, tens of thousands of cells at once to look at every single disease. We have been applying that to both cardiometabolic disease and brain disorder over the past 3 or 4 years. So for example, for a brain disorder, we studied traumatic brain injury. We analyzed close to 80,000 cells from 3 different tissues, two brain region and also the peripheral blood. So there are many different cell types. Each dot is a cell, okay? And each dot is summarizing thousands of genes in that cell, okay? And different tissues and different time points. I'm not going into detail. What I wanted to tell you is the power of the data that we could generate nowadays, and we can also use it to tease apart more detailed mechanisms in the disease. So for example, using the data, we could model how the cells communicate in the brain. This is the hypothalamus cell-cell communication map based on the data we generated. So this is in control animals. This are in animals with traumatic brain injury. You can look into more detailed interaction. For example, here between the astrocytes and neurons, these are the predicted interactions based on the data. And then in the disease situation, you can see the interactions are changing, and there are many different genes that are communicating between the different tissues. So kind of having more granular maps. So to summarize what we have learned here, this type of system biology can help us dissect the tissue and cell type disease mechanisms, and also can help us pinpoint the key regulators, and also potentially can help us find a new therapeutic agents to normalize disease networks. So the last 2 slides I'll focus on talking about the importance of bioinformatics and big data analytics in this area. Again, I bring up this particular slide to remind you the high dimensionality and the complexity of the data. So to do this kind of research, you do need highly interdisciplinary knowledge and skills. So you need to be able to design analytical algorithms and computing programs, and build databases and tools for yourself and for others. And on the system biology side, there are people who think they are the same thing, but I do not believe they are the same thing. I think system biology has a lot more of the biology component. In addition to bioinformatics skills, and also I want to emphasize even experimental capacity, because just modeling the data, integrating the data is not the final step. You really need to show in experiment that they do work. So basically, I think this image from the Institute of System Biology summarizes, right? It's a field that we integrate biology, technology, and computation. We have biological questions to address, integrate new technologies, new data, and new software to generate new hypotheses and insights. Then we go back to the circle to eventually get to the full picture. With that, I want to thank all the people who are involved in the few projects that I showed here. So I actually acknowledge the many of them on the slides when I introduced the project. Also acknowledge our funding from NIH. And my lab members here. So they definitely a very happy group of people, and we all, work, I guess, in a pretty pleasant environment, and really love their energy and all the contribution. Thank you, everybody, for your attention, and I'm ready for questions.
Thank you so much, Dr. Yang.
All right. Of course. All right. Let me see the chat. Questions? Do you take high schoolers? I did take one high school student so far. So yeah, the lab is a little bit saturated. So it's getting harder for me to give everybody attention. So my philosophy is that I need to give everybody who come to the lab enough attention. If they just come and I don't even have interactions with them, I feel like I'm failing them. So, if everything matches, and if you are very enthusiastic, we do have kind of senior student trainees who can help give you more guidance. I'm happy. And what do you mean by experimental capacity in systems biology? Oh, what I mean is you need to be able to do experiments, to actually design experiment to test what you found, and know how to do the experiments as well. So that's what I mean. Okay, what else? Has any of your work led to subsequent reduction follow-up work? Yes, so I showed in the fatty liver example where we found 4 predicted genes, and we went back to the reductionist approach. We take them out, right? Take the gene out in the liver and look for downstream mechanisms. And what we found was that they affect, 2 of those affect mitochondrial function. So we do go down to dig into the more detailed mechanism for individual genes. Yeah. With COVID-19, is your lab open in fall? We're open all the time because of this, mostly online, right? As I said, 70% of the lab is computational. So all you need is a computer. You can access to our big computing cluster, log in, and we have been working remotely from mid-March. So yes, the wet lab side has reduced the capacity for sure, but the dry lab is fully open. What are the biological or biggest challenge you face in leading such a diverse team? Actually, I didn't feel a lot of challenges. I find that it's a lot of fun to work with people with very different background, because they can, you know, everyone brings a very unique perspective. They can teach me a lot new things. I feel excited about it, and I don't feel like intimidated because, you know, science is constant learning, right? You learn, you improve, and just because they are your trainees doesn't mean, you know, they know less than you. Actually, many of them bring very new techniques here. So I actually, I don't feel very challenging. I do feel sometimes that I can't guide certain directions because I lack that knowledge. So I do do kind of try to build connections with more collaborators and things like that to help bring in more you know, expertise to help guide the trainee. Yeah. Can you expand on epigenetics? Epigenetics, it's so I guess it's more of a, I would see as a modification or reorganization of the genome. So one most common type, I don't know if you guys know, is the DNA methylation. So on the cytosine, on the C letter Cs of your ATCG sequence, you can add a little methyl group, and that will change the function. That will change whether the gene is going to be turned on or off. That's only one type. The epigenomic field is so broad that, you know, I can give you a full lecture on that. All right, see many more questions. I'm proficient in Java, Python, MATLAB, and some other. Oh, that's good. That's great. That sounds like you have a full package. If you know R, that would be even better. All right, and the helpful resources for students who are interested in this area of research. So there are a lot of online classes nowadays on bioinformatics, essential tools like Python, MATLAB, R, you know, Linux, you know, SQL databases, etc. So yeah, anyway, I think I'm taking too much time already. I don't want to go overboard. I hope I answered most of the questions. Thank you so much.
Sorry, give me a sec. Okay. Yeah. Thank you so much, Dr. Yang, for joining us.
Of course.
It was really awesome to get to listen to your talk about systems biology.
All right, great. Thank you. Thank you, everybody.